I am muted. There we go. Hi, everyone. Welcome. We're here with a few of us in person. Thank you all for showing up here. Appreciate it. And this is our first time in this new room. It's a beautiful room, but it's a wonderful place to have our both inaugural in-person session and Carlos's final in-person presentation as a Vanderbilt faculty member here before he moves on. But today we're very fortunate to have one of our own, Carlos Lopez, speaking today. Let me give a little background on his training experience. He got his bachelor's degree at University of Miami in Coral Gables. Went on to get a PhD from UPenn in Philadelphia, where he really embarked on the path that led him to where he is now, using computational modeling. Went on to do a postdoctoral training at UT Austin, doing a lot more in-depth physics. He calls it a lot of writing on paper type of physics, but he realized his passion was more in the biological side. So went on to do a postdoc at Harvard Medical Center with Peter Sorger's group. And that's where things really started taking off, and his work developing the PySB language, the Python-based computer modeling framework for biochemical network analyses is really did some seminal work developing that. So that led him here to Vanderbilt in 2012, where he joined the Department of Cancer Biology, which was dissolved a few years, how long ago now? 3 years ago, 4 years ago? 2016. Yeah, so it's been a while. So in 2016, switched over to the Department of Biochemistry, where he is now become a tenured professor, associate professor of biochemistry, and really looking forward to his talk today.
Hear me? Can you hear me? Yep, you can hear me. Good. So thank you so much for everyone for joining. And this is kind of a cool talk, and I was looking forward to give this presentation, because this is what I'll talk about today is kind of a culmination of the last 10 years of me working here at Vanderbilt. And so what I think is interesting is that sorry that I'm fumbling here. Anyway, so that a lot of times when I talk to people, they will say things like, oh, what kind of models can you do? And so I'm very interested in mechanisms and understanding processes and what drives these processes. And usually the challenge has been, well, what kind of data is there available? And did you already collect the data? And how do we get the data? And so it got me to thinking a lot about data and models and the relationship, and how the data that we collect informs knowledge in general. So I'll talk to you a little bit about today, about measurements, data, and models. It'll be a little bit philosophical at the beginning. I'll talk about quantitative data versus non-quantitative data, and can we gain knowledge from non-quantitative data? And I'll talk a little bit about this. How do we avoid the biases if we want to integrate data? And how can we use this to actually explore processes or specifically network-driven processes, which is what I'm interested in.
You can start your video.
I can? Okay. Do you want me to?
Sure.
Okay, you get to see me now. I don't see myself though. Is that okay?
Yeah.
Okay. Okay. So we'll go from there. All right, so let's start with this. So the traditional way that we were taught, I guess, at least that I was taught to build models, was you start with your model, you start with your system that you're interested in, and you scour the literature. Usually this takes days, weeks, months, and usually you build something that kind of captures the knowledge that you believe is true. So in this little thing here, I'm just showing a little cartoon of, for example, apoptosis, which is one of the model systems that we've been very interested in because it's very amenable to modeling because it's a very clear outcome. The cell died, and no one's going to argue with that. Usually, people can, and then and it's also very important in terms of health and disease and all these things. So we build this dynamical model. We describe a time-dependent process, and we encode this, right? And then usually what people do is they calibrate the model. So you take some data. In this case, it could be fluorescence data or some quantitative data, and you just fit your model to some function, for example, some sum of squares of the experiment relative to the data, and you calibrate the parameters of the model until you get the model to agree with the data, and then you use the model and start to uncover things, right? But the challenge with this approach is that, or at least one of the challenges with this approach is that when we do this, we're taking usually these very big models, and we're just minimizing this distance. And then, you can also do it in a Bayesian way. I'll talk about this in a minute. But it requires a distance metric. It requires you to have a metric between your model and the data and what you really mean, right? And this is where you start to assume things. And this is where you start to say, okay, well, I'm going to assume that this signal in my model represents this, and I'm going to assume that this measurement in my model represents something else. And so, you put all your assumptions, you fit this thing. But what really happens is this: you end up with a model. Typically, the model, if you want it to be truly mechanistic, you'll have tens to hundreds of chemical species. You'll have hundreds of chemical reactions. But it turns out that you only have maybe 2 or 3 data sets or a few data sets that do not constrain the model completely. So you end up with an underdetermined model. It's a model that the parameters are either poorly identified or not identified at all. And so what that means is that what I'm showing you here is 30,000 fits of a model to the same experimental data in apoptosis execution. And the problem with this is that you cannot tell which of those vectors of parameter fits is better or worse than the other. And so what people usually do is they say, well, we're just going to choose one, or we're just going to choose a few, or they don't even look at the multiple parameter fits at all. And the challenge is that each of those could give you different dynamics. And this is what we're after. So we started to ask 2 really more fundamental questions. The first one was, how does the data inform the model in a way that tells me something about what I'm looking at? Number one. And number 2, how can I extract mechanisms from these things that actually guide experiments? So that's what we started with this. So we departed from this kind of traditional view of fitting models like this, and we turned to a more Bayesian model calibration to kind of, and you can say that we like to think that Bayesian calibration allows you to reveal how informative the biological data are. And what we mean by that is, or what I mean by that is, instead of getting one parameter vector or one value, I start to get distributions for each of these parameters. And so each of these distributions is going to tell me, well, some parameters are very well constrained by the data, and some parameters are very broadly defined by the data. So I can at least start to understand where the data is constraining my model, or where the data is being very loose with my model. So this is a little bit better. And then what you can do with this is we obtain these parameters by basically optimizing this kind of function or sampling this kind of function. I'm not going to go into details, but it still gives you hundreds or thousands of equally good fits. But at least now we're sampling what we would like to believe is the complete, or as close to the complete, probability distribution of the parameters. If you can get these models if you can get your parameters to sample enough of that parameter space, you at least have some understanding of how your model and your data are constrained, or rather how your data constrains your model. But then this gets to another problem, which is, we're used to this idea of data that can be directly related to a model. And what I mean by that is, so I took this from the web. If you have a train that, well, we're here in Nashville, so if we want to go to San Francisco, we're kind of, screwed because it's over here. But if you're here in Nashville and you want to go to San Francisco, you can measure all these distances here and track your path, and that time from very simple equations that the time it's going to take is depends on the distance and on the speed that you travel. So if you know the speed of your train over here, I chose this Amtrak. I don't know which ones come here. But if you know the speed of your train and you know how far it is, you can just plug those 2 numbers and get time out. And we're very used to this data that can be very much plugged directly into your model. And this is very much an engineering or very physics way of thinking about it. So the challenge I find, or we found over the last 10 years that I've been here at Vanderbilt, is how do we think about data better? And so we started to think about, how informative is data and how do we deal with data that is more or less informative? So we started to think about, how do we classify data? How do we define data? So we started to think about, different types of data. And this is work by Michael Irvin. And so you can, and the question, the problem is that in biology, we can ask questions that are different. And so the data will inform the question in a different way. And so to my little example here, I think everyone that knows me knows that I love coffee. So the question is, do you like coffee? And you can have a nominal answer, yes or no, or meh, that now you have three measurements or three metrics, right? You could have an ordinal measurement where it says, 1 star, 2 star, 3 star, but there's no clear, metric in the stars. It's just a star system. And, sometimes 5 stars means it's great and 1 star is terrible, but what about 2 or 3? Is it meh? And how do you define 2, 3, and 4 and the differences between them, right? You can then change your question. And so instead, do you like it to, do you drink a lot of coffee? And the answer could be nominal, yes. It could be ordinal, a little bit or a lot or medium. And it could be semi-quantitative. How many cups of coffee a day do you drink? And you can make it absolutely quantitative. You could measure exactly how many milliliters of coffee you drink a day and actually put a number into this question, right? So we see the same kind of trend in biology where we will be asking things like, what activates a process, or how do you kill the cells? How do you do these things? And the problem is that how we pose the question, what data we have, might give you different answers. So now with this thing, we said, okay, well, we can identify at least 4 data types. There's a nominal data type where, it's either blue or green, died or alive, some phenotypic state of a system, right? There's a ordinal data, like something like like a Western blot, where you have a little bit more, a lot more, and there's clearly an order to the data, but you don't know much about, you don't necessarily know the quantitation. There's semi-quantitative data, which is basically data you can measure, like fluorescence data, which is a function, hopefully linear, or some sort of function of the observable that you're actually interested in, like the concentration or some number. And there's of course quantitative data like mass spec, where you actually get the number and you can plug that into your model. Okay. So the challenge with these data sets is that they inform your models differently. And this is where I'm very intrigued about how to work this out. And so what people traditionally do is they define a scale. They say, okay, well, if I get this kind of fluorescence, I can map this to how many molecules this fluorescence means, and I can make a function that maps these measurements to what I want to know. In this case, maybe protein concentrations, and I can get maybe a pretty distribution that where I can know where these things happen, right? But things get a little bit more complicated when you do things like Western blots, right? Because how can we define a scale and intervals for relating this? And so are you going to say, where do you put the cutoff? So this might be a very broad distribution. This measurement might map to a more narrow distribution. And the problem is that the noise obscures those intervals. And at the same time, we want to use as much data as we have. So what if we wanted to integrate the data? How informative is the data? And how do I classify these things from different data types? So we decided to pursue this problem, kind of in a different way. So what I'll show you today is, how do we incorporate non-quantitative data, unquantitative data for mechanistic models, and how do we understand basically where to draw the lines? So we have a measurement that looks like this. Where do we put the line? Do we put it here? Do we put it here? Where do we divide things? Or could we actually just, express it probabilistically? And you see where I'm going with this. We're going to express it probabilistically. And so what Michael Irvin, was he designed a tool that basically lets you machine learn. It uses a machine learning approach to classify where the data belongs in its classifiers. It finds classifiers for the data, and then it lets you see how it maps to a model. We're not the first ones to come up with this idea. The people at PyBio and NetFit used ad hoc definitions of the qualitative data, for example, equalities or inequalities. And PyPesto also did a great work that uses optimal scale thresholds, but none of this did it in a kind of Bayesian probabilistic way. So that was kind of our innovation. We brought a machine learning approach and a Bayesian approach to really kind of classify and understand how the data and the models relate to each other. Just again, please check this out. These tools which are really cool also, and they're very applicable to many other problems. So I'm not trying to badmouth them, because they actually were a huge inspiration for us. But to point out that they exist. The BiBenefit has Bayesian, a Bayesian approach, but it requires ad hoc assumptions about the measurement models. PyPesto is data-driven, but it's not Bayesian. So we wanted to build our own. And so what we introduced here, what we're trying to do is this new idea that rather than taking the data and putting it straight into the model, we're going to put something new here that we like to call the measurement model. And the measurement model is basically another little tiny model that you add to your system that interprets the measurements in terms of existing knowledge and helps you map that measurement into your data. And so what does that mean? It means that, so if you know you're mapping directly, if you know that this mass spectrometry measurement is directly a number in your model, then you don't need this, right? But if you have a measurement that is a fluorescence measurement or a Western blot, and you want to map that into your model, then this is going to help you understand, how to classify and where this belongs in your data. This adds a little bit more computation, but we believe it lets you better understand how your data is constraining your models. And I'll show you what I mean by this in a minute. So, okay, so this is essentially what we're doing. We've redefined the likelihood function to include, of course, the number of simulations, the category of the measurement, the probability of data being in one category given the modeled category, and the probability of the measurement category given the simulated value. That's it. So we've basically expanded the traditional Bayes equation into something a little bit more complicated. The framework, I think, is fairly straightforward. The main idea is that we follow this algorithm where we draw dynamical and measurement model parameters. So the theta are the data parameters, are the model parameters. No, sorry, the dynamical parameters and the measurement model parameters. We draw them from some prior knowledge. It could be if you know a lot about your data, you can impose those constraints in your system. If you don't know much about your data, then you don't have to put constraints in your system. We simulated dynamics of using some solver. In this case, we did ODEs, but you can use stochastic differential equations or any other solver that you're interested in, Boolean. Predict the probability of each measurement category using the measurement model, and then we evaluate the likelihood. And then we use the Monte Carlo Metropolis Hastings kind of sampling to draw new parameters from their priors and keep going until you get some convergence. So we've added a little bit more complication, but again, now we're not biasing where the measurements belong. The approach simultaneously calibrates the dynamical and the measurement model data, and it can be generalizable to many other model systems, and we actually have shown this. So, okay. So now this is the math part, and I'll show you, I think, hopefully the fun part that I hope everyone will enjoy more. Well, I enjoyed this a lot too. So the first question was, how much ordinal data do we need to attain comparable certainty as semi-quantitative data? So we built a small model I'm showing here of apoptosis execution. The way this works is very simply that you have a receptor and a ligand, in this case trail. They make a complex. This complex can activate or induces the formation of the death-inducing signaling complex. This disk can then activate the initiator caspases. The initiator caspases then activate BID. BID gets truncated, goes to the mitochondria. In this case, we abstracted all the BCL2 regulation into what we call a MOMP signal, a mitochondrial outer membrane permeabilization signal, where we say it's off or on, and so pores are made. Once this is on, the effector caspases get activated, and the cell and PARP gets cleaved, and the cell dies. So we try to keep the model still biologically relevant, but make it as simple as possible because we only had this data set. This is a data set of, in the x-axis, I'm showing you time in seconds. On the y-axis, I'm showing you normalized fluorescence data that corresponds to bit truncation. So the only data we have is from experiments is at what rate is bit truncated, in a given cell. And this is average data from, I think, 100 cells, something like that. And this is data from John Albeck and Sabrina Spencer. And apologies for not having the citation here. There's two papers. I'm happy to give to anyone who's interested. Anyway, so this is our data, and this is the error bars. And so, of course, if we calibrate the model, and one more thing, we chose this model. We chose to simplify the model because this was the largest model that we could actually build where all the parameters would converge for just one data set. So we wanted to make sure that all the parameters would converge for this exercise. okay? So we did this. We calibrated the model to the data. This is what the calibrated posterior looks like. So remember, the data with the error bars is like this, and the dashed line is the true experimental data, and the solid line is the fit, and the gray area is the posterior, the 95% posterior likelihood, if you will. So of course you get a really good fit. We have a lot of data, we have a lot of quantitative data, and you get a really good fit. And we said, okay, well, let's pretend now that we have Western blots. So we just basically took the experimental data and we made synthetic Western blots by basically coarse graining the data and taking points at different time intervals, just like we do an experiment. And we added different kinds of noise onto the data, as you can see here. And so we did that. And in this case, we added 13 measurements. So typical 10 to 13 measurements. And again, here you can see that that the experimental data is here. This is our best fit, and this is our noise. It's very noisy. I don't think I've ever seen something like this measured for these kinds of models, where how certain is your model. Of course, if you add more data, things get a little bit better, but maybe not, because it depends how many time points. This is 60 time points that are distributed in different ways, and you get this kind of a measurement that again, you get more accuracy but less certainty. Now we go to 100 time points, where someone spent a lot of time measuring these things, and you get again better accuracy and better certainty, and so on and so forth. And it's not until you're at about 300 of these Western blot measurements that you start to recover data that is comparable, or fits that are comparable to the fluorescence data. So this was very interesting to us, because many times you will see conclusions drawn from models fit to data that may not be that may not constrain your system that well, and so this raises a lot of questions. Can we? Okay, so it seems like non-quantitative data can be really useful. And so the question is, can we improve certainty from non-quantitative data integration? And so we did a second exercise where we basically said, let's just focus on nominal and ordinal data. Let's pretend we don't have quantitative data. Let's pretend that all we have is what exists in the literature, something that was published 50 years ago. Can we use this? And so the answer is, we actually can. It's really interesting that so we took the same thing. We took the experimental data, and this time we made it into nominal, which was like how activated disk is, and we just made 4 classifications, and we played with different classifications, but just for the argument here, this is what we did, and we optimized the model to this data. I was very surprised that even though there's no numbers here, there's still mechanistic information here. Yes, this is the true data, and this is my prediction. But qualitatively, we're not doing too poorly. We're still seeing T-bit activation and all these things. So we said, okay, well, presumably we already know this, that we can do the same fit, or the same kind of fit with ordinal data. But the interesting question was, well, what if we combine them? And I forget what the measurement is exactly for the areas, the gray areas here, but it's about 50% reduction when you combine the non-quantitative and the quantitative data, and let the machine kind of classify these things and give us parameters. Of course, it's a poor fit compared to the experimental to the quantitative data, but there's information here, and that was the exciting part for us. So then the third thing we said, okay, well, so what people typically do when they do a Western blot, or they do something like that, is they take their Western and they assign a constraint when they put this data into their model. And so the question was, how does this affect accuracy and precision in our predictions? So to do that, we again went to the discretized data, and this time we actually created different kind of classifier bins as priors. And so we did 2 ad hoc cases: one where each of these measurements belong to one of these distributions, and another one where we just kind of changed the distributions a little bit and added more classifications. It's a completely arbitrary decision because there's really no way to guide this, right? And so in the first case here, we actually see that the model has a really nice accurate fit to the data. But in the second case, the model does much less well. It's a lot poor, a much poorer fit to the data. And so this starts to raise the question of, how much bias are we introducing with these things? So so we said, okay, well, what if we probe what kind of knowledge we have? So we said, okay, instead of doing this ad hoc classifications, let's start with different prior knowledge. So rather than impose the constraints, let's see if this approach will let us kind of learn where things go. So so this is when you have uniform priors, and so again, we had the data started in some classifier, and then this little distributions here show how the distribution of that data and that classifier moved throughout. This is how the measurement model started and finished. So that means that we learned something from the data, or at least that's how we interpret it. And so we saw that, with having just assuming that there's no prior knowledge actually gives us some good predictions, and it was actually really good at finding this. We then tried Akashi priors, which are basically so Akashi prior of s equal 0.05 just means that that we have some knowledge, but it's not very strong knowledge about the system. Again, we find that it fits things pretty well when you make few assumptions. These are less assumptions. And now this is the Akashi prior s 0.005, which means that we know a lot about the system, and we decided that we really know where this thing should be classified. And the interesting thing is, again, that this actually added to our uncertainty. So we're less accurate when we do this. So we're, again, the idea here is kind of a proof of concept to kind of think about, how is my data informing my model, and how are my biases, the things that the biases that I'm putting into my data affecting my model or my model prediction. So what can we do with this? So what we have now, okay, so let me just kind of recap a little bit here. What we have now is we have made a measurement model that allows you to take data of any type, quantitative, semi-quantitative, ordinal, nominal, and basically machine learn where it should belong, or where it actually informs your model, and you can deal with these things in the code. And so I can now measure how accurate my model is given the data that I have, and that to me is the exciting part. So then the question was, well, can we guide experiments? So we did another kind of proof of concept thing. Where we said, okay, what if we wanted to identify what would be an informative measurement? Let's pretend that we could measure, in this case, we said, let's pretend we can measure something that's related to the outcome, like maximum bit truncation rate versus the time at bit truncation rate, or two completely unrelated measurements, protein amount at maximum bit, some completely unrelated measurements. And so we're still working on how to use this better, but the main take-home message here is that this measurement is informative, and this gray region here tells me that this measurement would actually help me improve my prediction, the predictive power of the model, but this measurement would say nothing. And so the main idea is, when I talk to someone like Vito, who's a great collaborator for us, and we say, what data can we collect? Now I have a tool where I can say, with this simulation or with this approach, I think that this would be informative measurements, and this would not be informative measurements. So let's focus our attention in this area. And that's where we're going. That's what we'd like to do. So, okay. And then the last thing I wanted to kind of share is, well, we go through all this work, we go through all these things, and why? What can we get from this? And so what we are after really is, and I think many of us are after here, is how do we perturb a network to get a different outcome? How do we actually change the network to give a different outcome? In cancer, how do we perturb the network to have the cells die, or how do we reprogram the cells to go to a different outcome? And so these are all questions about the dynamics of networks, which is a really hard problem. But I think we're getting at something that is really cool here, because now that we can take these models, calibrate them to data, and I'm showing you work by Oscar here, where he used an ultra discretization / tropical algebra approach to understand the dynamics. We can now take that data and say, well, your model that was fit to the data, that we understand all the parameters in the space, only really works in kind of three execution modes. One where the signal goes through MOMP and goes down here, one where the signal kind of stays stuck in MOMP regulation, and one where it just kind of goes straight without doing this loopy thing here. And depending on which one you believe, you can assign probabilities to each of those modes, and now you can start making predictions. So, for example, if we were to target something here at the MOMP level, or something here at the QSBC level, would that be enough to guide my cells in a different way? And this is, again, just a proof of concept, but we can now identify these execution modes in the networks. This is what this looks like. If you take one parameter set and actually kind of find the quote unquote typical execution mode, here I'm just showing you a simulation where the bit is getting activated. And then at some point, you're going to see that backs gets truncated, and then execution of the apoptosis signal takes place. And this is when the cell dies, right there, the cell died, it's over. And so that's one way in which the signal was executed from one of the parameter sets. And then I can do that for another system where, it's a completely different parameter set, and I can see the mechanistic interpretation of how, this one's more dependent on disk formation. And again, the mechanism is a little bit different, but it is what the data tells me. So, we can start to compare these mechanisms and find things like, what are the differences, or what differences do we expect from these mechanisms? And can we actually narrow this down to improve our predictions and to guide our experiments? So this is where we are with this kind of work. So we're doing questions and answers after this, right?
Yeah.
Okay, good. So I'd rather do more questions and answers then. So just to and I get really excited about these things because I think that at the core level, we like to make all these measurements, right? RNA-seq, singles RNA-seq is such a everyone wants to do it right now. But how informative is that to the data, to the question that you're asking? And that's where we're getting at. How informative is the measurement to the question that you're after? So I just showed you, we introduced this concept of a measurement model to integrate different data types to model biological processes. It's not a significant increase in computational resource. It allows you to remove biases in your optimization. We can now explore mechanistic information more broadly with available data and understand what informs the data more or less. And then now making in silico experiments to guide measurements, I think is something that we can do. We're going to be able to do in a more directed way. This is the product of many years of work. So I just wanted to highlight some of the papers that kind of have touched upon this a little bit. This one's still in review, but I'm thinking it looks like there's light at the end of the tunnel. I'll keep you informed. And of course, some of these papers are all in bioRxiv. And then I'll stop by, since this is my last talk as a faculty here at Vanderbilt, unless there's something between now and Friday. And I've had a wonderful time here. It's been a great 10 years. Vito, it was 10 years ago. Vito's here, he brought me here. I have nothing but good things to say about what happened here. I've had great colleagues, and it's been a really fun time. And we've done a couple of great things here, and we're going to continue. For those of you who don't know, I will be moving to Altos Laboratories starting Monday. So as of Monday, I'll be an employee in the West Coast, although I'm starting remote. And then of course the people that I really have to thank is my lab, past and present. The work that I showed you today is mostly done by Michael Irvin, who's now a research scientist at Argonne National Lab, and Oscar Ortega, who is now I don't know what his title is, but he's working in applied biomass, and he's having a lot of fun. And of course, there's other members of the Lopez lab, and there's the babies of the Lopez lab, and I'm gonna miss everyone, and I'm gonna miss you guys too. So but now I'm in the West Coast, so please come visit. And I'd rather answer questions and talk more about this because I know that I planted a lot of seeds of potential discussion here. So thank you. And I get an actual clap instead of the virtual,
I got a question. How should we do this?
I don't know.
Go ahead and just say it, and then hopefully Carlos or I will.
So this idea of essentially a data pre-processing model.
Yeah.
So there's always the problem that data is usually not even in the same units as the very model just to begin with, fluorescence, for example. So when you're thinking about these measurement models, you kind of pitch it almost as going from some sort of nominal or ordinal to continuous data. But I guess the first question is it restricted to that? And the second question is what kind of form are these measurement models taking? Just kind of like small people.
Yeah. So both great questions. And so, okay, so the first question was, you can? Okay. Our sound engineer is telling us that we're okay. Okay, so okay, so we started with a very simple, logistic regression classifier. And so what that has allowed, and we're assuming that there's that the data could be continuous or non-continuous. And so you're right. The question, the challenge is how do you map non-continuous measurements to a continuous space? And what the machine, what the tool has allowed us, with this classifier, what it lets us do is identify where that parameter, kind of maps or has the most weight in the model, right? It could be fancier, and we started very, very simple. For example, we've thought a lot about things like regularization and how do you deal with that, or feedback. How do you deal with those things? We haven't gotten there yet. This is one of the reasons we started with a very simple system, but that is kind of in our radar. For the other part of your question, which was, how do I envision dealing with different units and things like that, or different measurements? I don't know yet, but so far right now, what we've taken is data from experiments that we knew or from collaborators that we know, and we understand the data well. So from where we are, from this kind of proof of concept idea to actually an automated thing where you just press enter and everything gets classified for you, we're not there yet. But you're right in that's where I'd like to go, and so that's going to be one of the big efforts I'm going to be working on in the next 5 years or so. But those are really good points, and that's something that I very much want to do. Yes, go ahead. Thank you.
About the Western block and such into the data, how do you deal with the general messiness of data that was collected in the lab? And how do you, or are you thinking about what is it? Like, is this noise driving your signal? Is it taking away from your signal? And what are some things that you've done to address this?
So, okay, so I'll answer the second one first. So, depending, so I didn't talk about the structure of the model, but that's something we're also considering, So it turns out that depending on how you do the machine learning bit, and how you specify your priors, you can increase or decrease the noise in your system. So now it becomes an optimization problem of, what is the optimal way to optimize the data with the least amount of noise, and it's just basically more computation, and there's ways to do that, and in fact the work that Sam has been doing is kind of related to that. So that's one thing. The other thing is, how do we deal with the messiness of data? So right now, for this part of the project, we've dealt with data that we understand very well, and we have taken data that we know. So for example, Western blots, we knew how they were taken, we know what they meant. We actually did synthetic Western blots for part of it as well. I would imagine that for the future, for when we start to incorporate more real world data, there's going to have to be some manual creation, at least at the beginning of what this looks like. So I don't think that, at least initially, I don't think that this is going to be a fully automated process. I think it's going to be more of a human in the middle process where there's some rounds of optimization. You kind of pick the parameters or the classifications that are useful and informative for you, and then you let the computer keep going. But even that, I think, would solve a lot of challenges in terms of how predictive is your model. Because and this is answering a huge problem in our field where there's kind of two camps. One where the bigger the model, the better, and we're going to explain everything. But then the other camp as well, but you don't have that much data, so how useful is your model? And that's exactly where we want to get. Does that answer your question? Okay. Anything else?
Any online question?
Oh yeah, nothing. At least not that I see. Hey, online people, go ahead and ask. It's okay. Yes, of course.
Sample the data you said you did some deep the synthetic data set by artificial sampling. So was there a lot of bootstrap involved, or did you?
Yeah. So we did several things. So for example, what if we do even time intervals, or what if we do time intervals only when the changes in the data are more drastic, or things like that. And it turns out that the most useful thing, and we can talk about this more. I didn't want to talk about this. So we did a little bit of dynamic time warping to kind of understand how to sample the data. And it turns out that really what you want is, which is kind of obvious, is you want the most measurements around the most important dynamic changes. So if you know that there's going to be a dynamic change, then, you know, measure more at that time interval. However, we don't know that a priori, so, you know, the most data you can give me, the better. But, you know, but what we found also, and this is data that we have not published, what we found is that when you start playing with this prior knowledge, when you start playing with this prior knowledge and you have strong belief that there's knowledge that really impacts your model, then you have to actually impose it on the system. For example, in some of the work that we've been doing with the parental lab, we've been looking at tumor growth mechanisms, tumor growth dynamics. And if you just let the system kind of learn the best model, it tells you, oh, you know, on and off, you know, that's all we care about. But we know from experiments that there should be at least 4 subtypes. So we have to kind of hard code that into the model because the computers are stupid. They just want to give you the simplest answer. So there's something about that aspect of the data processing and the prior knowledge that impose into your system. And that's where expert knowledge comes in. And, you know, Sam will be talking about this soon too. So there's a question online. Jason asks, how do you distinguish between any uncertainty in your underlying model structure and interpreting the output of the measurement model? So what you can do, and that's a great question, what you can do is basically, you can do analysis like adding or removing perturbations into your system to see how it affects your measurement. And because the measurement model is an actual model, you can actually change the parameters in your measurement model to see how that affects your output. Sorry, and so about the choice of topology, that is, so I'll be happy to tell you more about that, but it's a whole other, you know, hour conversation because it's a really complicated question. But essentially what we have done is we've actually used approaches to measure the Bayesian model evidence and then compare multiple models and see which models take more or less probability space. And I don't know if that answers your question, Jason. And then Laura Richardson asks, can you talk about bias considerations in these models? Is there bias in machine predictions and how would it compare to human analysis error? Also, okay, so I'll answer that first. So what I'd like to see, or what we see, is that, and this is exactly what we showed here, that, you know, human biases can either be really good and give you a really good way to classify your data, or really bad and you know, really mess up the predictive power of your model. So you don't know a priori which one's going to be. Also, would the model be able to discern data that really doesn't make sense? Yes, that's one of the things that we're trying to do. So the measurement model could help you understand why some data is not informative and some data is informative. It could be that the data is just not informative at all and you're assigning value to something that's not informative. And then the last question is, I also wonder, how do you see this type of work being integrated into BioLab? How much training would it be needed to operate this model? So right now you would need some training, or at least working with someone in my lab. We would like to get this to the point where it's more automated and more turnkey. We're far from that. I mean, this is really today proof of concept kind of ideas here, but we would like to automate this in a way that, you know, what I would really like to do is you take your data, you build your model, you optimize your model and your data, and you get optimized parameters and how certain your model is given your data, or how predictable, what is the predictive power of your model, given the scope of what you're looking at. And that's kind of the vision. So I think you, at least, was this Laura? So I think, Laura, you're kind of, you understand what I'm trying to say. So stay tuned. Yes.
I'm gonna
Yes, of course.
a bit of a pessimist.
Okay.
So my understanding is that in a perfect world, you have nice kind of continuous good data. You don't have a perfect world. Your model produces this nice continuous good data, but you have a compressed version of that coming out of Western model.
Correct. Yeah.
So what you're effectively trying to do is you're trying to learn an optimal compression of your model into your data space. And this kind of gets into the summary statistic problem, which is well known that anytime you start compressing data and compressing models, if you do not meet what's called like this sufficiency condition, basically if you have a bad compression, then there's nothing you can do about it.
Correct.
Now you're trying to determine compression that is suitable given the compression of the data, but what if the data compression itself is just bad?
Absolutely. Yeah, so that is a great point, and it's something that I spend a lot of time thinking about. You know, in other words, if your measurements are Yeah, if the compression of your measurements are not are poor already to begin with, then you're going to get crap models. And I don't think that there's a solution to that yet. But what I will say is I think that we have a lot of work to do in the field in terms of understanding even how predictable our models are. And I think that having a handle on that would be good. I think that the challenge with understanding, like you said, in a sense, what you're saying, what I'm understanding is you have this measurement, and what is the relationship of this measurement with the knowledge that I think I have about the world, and is it good or bad, right? And so I think that traditionally, and now I'm gonna go back to Einstein and Newton and these people. Traditionally, what has happened is you kind of believe that model until it breaks, right? And so if the model, I mean, this are all empirical models, right? So if the model works and it gives me predictions, then I have a reason to believe in it. And the example I can think of is Newton, right? So gravitational theory, everything works great, except if you're really big or you're going very fast, now you need relativistic corrections. And so I think that we are not at the Newton model yet. In fact, we're not even at the Kepler model yet. Like we are, I'm sorry?
We're still on that cycle.
Exactly. We're more or less Copernicus here. And so, but you're totally right that it is very possible that at some point that we come up with a model that explains, I don't know, aging, which is what we're going to study in Altos. And then something happens that breaks our model and we have to revise everything. But we're not even at the point of epicycles. We're kind of scratching the surface. But that is a really good question because, and I don't know the answer to this, it gets to the point of, I think that many times we do measurements in biology because we can. So, 20, 50 years ago is Western blots, then, fluorescence came along and it was all about tagging things. And now we have all these omics techniques that are really awesome. And we do them because we can. But I'm willing to talk about this over coffee or beers with anyone. There are times when really well thought out Western blots are more informative than 10 proteomics measurements. And but we don't know what that looks like yet. And I think that this is opening the door to explore those questions, but we're not there. And that is, you just got me going. So I'm going to stop here. But this is something that I think a lot about. This is the kind of stuff that I think about when I'm falling asleep. So, but I'll leave it there. Is that okay? Yeah. But that's a really, really good point. Because just to add one more thing, in physics, for example, in astronomy, they understand the compression of the data and what it means really well because their models are very strong. And so when you break that model, you know whether it's your data, whether it's your model. But in biology, we're not there. And that's I think the philosophical kind of challenge we have. Sorry, I just wanted to add that part. Anything else? Okay.
Proud of that. That's I mean, the type of stuff you're going to be doing in Altos is much more complex. This is a very simple question, essentially. Here, in this model system, all the cells are dying. You're just measuring how fast they die, essentially.
Yeah.
And aging's much more complex. And how are you going to, what's your vision for applying this modeling to such a broad question, and how are you going to be able to?
So I don't have the answer to that yet, but what I do know is that we have a lot of resources to collect data that could be more informative and to build models that are more complex. But I don't know if more complex models are going to give us the answer. I think that there's kind of two camps, let's make the model more complex. And then there's the camp of, let's make it all exponentials and call it a day. I think the question is more, how do you model the data to answer the questions, right? And I think that's the fundamental challenge that we don't know yet. And I think that because the problem is inherently multi-scale, how much molecular information do you include? How much cellular information do you include? How much organism information do you include? How do you integrate models? How do you integrate all this data and models to give you predictions? I think all eyes are on the modeling team because of that, because I don't think there is.
That's why I was curious.
Yeah, no, but it's actually, but it's a very valid question because the answer is no one knows. And that is why that's why they put this team together, because they think that we at least have a chance of maybe, starting to answer those questions. I know we're going to start with not very simple, but we're going to start with a simple problem of homeostasis, proteostasis. How do cells maintain a balance after given a perturbation? And what is the machinery involved in maintaining those perturbations? And why is it that, young cells recover quickly, but older cells may not recover? They will go to another state and stay there. And so, but that gets even more fundamental problems, like how do you define a cell state? What is the identity of the cell? And you can look at the microscope and see the cells one type or another, but if you could take RNA-seq, which is our state-of-the-art measurement, is that enough? And so there's a lot of fundamentals that we're going to try to fill the gaps in the next few years. So stay tuned. I'm excited. Anything else? Yes. Yes, go ahead.
So on the Untitled Posterior for the parameters, that my understanding was you said you get different uncertainties for, let's say, structural parameters. Are there, is there a specific relationship between the mutual parameters that describes the state? If you have
So we haven't looked at that. So there should be, you're right. So if I understand your question correctly, different combinations of optimizations might give you more or less uncertainty in the parameters. And if is there a correlation between the uncertainties? So I think that some of that we kind of touch on that indirectly in the kind of the prediction of the dynamics of the network. But I haven't thought about it in terms of, kind of the shape of the variance space or something like that. I'd have to think about that more, but that's a really good idea. I would imagine that you could. No, I'm not going to say clustering, but but yeah, I see where you're going. I don't know. We've been more focused on the dynamics of the network itself. But I would imagine that the correlations between the parameter sets that make these different modes of execution, there would have to be some correlation in their noise also. I don't know that off the top of my head, but that's a really good point.
I have follow-up question here. That was
Are you a reviewer of our paper? Because I feel like someone asked us that, something similar too.
there are natural variations.
Right.
So
Right.
If using this inferred models that you have given this data, if you have looked to say, let's say you have this observational set under this one queuing condition, if there's a way that, let's say, we can expose the cells to different conditions, and what would model say about that?
Right. So can we break the model? And I'm sure the answer is yes. So like I was telling, Bill. But to answer your other point about the variability in the proteins and stuff like that, since you seem interested in how much variance is there between different measurements, there's a really good paper by Stephen Wiley in Science Signaling. I think it was a year or two ago. I don't know if you know this paper. And he looked at protein variance under different kind of points in a network. And what he found is that the kind of hubs in the network are much more tightly regulated in terms of gene expression than kind of the spokes of the network. And so he did a very interesting observation that because people usually believe that the coefficient of variance in proteins in a mammalian cell is 0.3. And so, 0.2, 0.3 from cell to cell. But what he actually showed is no, it depends on the function of the protein. And so it alludes to what you were saying. And that's very much in our radar, but we haven't gone there yet. But that's a really good point. Yeah. Thanks.
So before Nito goes in, there's one more question.
Oh, okay.
Chen asks, how could the type of model you capture emergent?
So one of the things that we were trying to do is so, for example, the work by John Albeck, who talked last time in the same venue. We've been talking a lot to him because one of the things he sees is that when you perturb cells and induce growth or whatever, when you look at the single cell behaviors, they are doing such different things and they're all over the place. But the emergent behavior of the cells growing is very much, you can measure that and you can count how many cells are growing. And so one of the challenges is, if we really do believe this networks and we believe that they actually make sense, can we capture the behaviors of those individual cells with the same model, but with different parameters, obviously, and then estimate how much each of the cells is contributing to the population behavior. That's work that is kind of ongoing right now. I love these questions because it's exactly the kind of stuff that we've been thinking to move forward. But I think this is going to be the workhorse in answering those questions, in kind of saying, how much does one mechanism versus another mechanism contribute to the overall observed behavior? That's where we're going there. And in fact, and I left it. It's good that it's here because this is kind of teasing at that. I'm not saying that one of this is the right one and one of this is the wrong one. I'm saying that the cells might be executing signals differently, and they might respond differently because there's different ways in which the signal could be executed. And so I think that the better way to think about mechanisms in cellular processes is probabilistic rather than deterministic. And that's kind of what we're going with this.
What's what we are asking. Last question.
You are.
Another question. Statement. Constant to bring up in the 10 years.
Sorry.
So 10 years go by fast, by the way, very fast. And so you think back and you say things that we should have done, things that we shouldn't have done. But the bottom line is this, and the colors make the difference. And so I'm thinking about, these 10 years without colors could be very difficult to get a difference in many more ways than one. First of all, I think the difference in itself, he discovered to be a theorist, basically with very different ideas.
Yes.
It's a crowd from the top today, that is really gone pretty deep. Deeper.
It was not my intention. It just, was the answer that was there.
And then it made a difference to my lab, certainly. It made a difference to the department. He also made a difference to a bunch of people that he brought here. So one of the towns that Lopez is really, again, really select very, excellent people, outstanding people. So if you got everything Carlos's lab, you can be proud of that. And so I mean, what can I say? Then on the 10 years, you become friends.
Yeah.
Fortunately or unfortunately. So we did good friends, and I think we would stay.
Absolutely.
And, what can I say? I'm elated, that you got this.
Thank you. Thank you.
Well, we're going to be.
Thank you. No, it's an excellent opportunity, and it's always mixed feelings, right? On the one hand, I'm gonna miss you guys, but on the other hand, there's a really bright, shiny thing on the other side. So I'll report. Who knows? Maybe in 5 years I'll be asking Bill for a postdoc. So we'll see. I need a job or something. So yes, exactly. Yeah. Thank you, Vito.
All right, thank you. We're gonna close the session here. Thank you all for attending. All the students and postdocs are welcome to join on our continued discussion in the chat or in the new Zoom that I just posted. Thank you.
So how does this work?